Hello, good morning. Myself, I am BSR Botany lecturer. Today our topic nucleic acids. So, nucleic acids are macromolecules, whereas nucleotides are micromolecules. So, these macromolecules, nucleic acids, first reported by in the nuclei of pus cells, mainly by Friedrich Miescher. So, the nucleic acids first reported by Friedrich Miescher, where he discovered in the nuclei of pus cells. The term nucleic acid was coined by Altman. Nucleic acid was coined by Altman. Here the nucleic acids are two types, DNA and RNA. You know DNA containing deoxyribose sugar. This is containing the deoxyribose sugar, whereas RNA containing ribose sugar. You know that one. So ribose sugar formula C5H10O5, whereas here ribose sugar C5H10O4. So here DNA, first we are discussing deoxyribose nucleic acid. Then we will go for RNA. That DNA double helical model proposed by Watson and Crick. Double helical model proposed by Watson and Crick. Remember that one. And mainly transduction experiments. We are discussing purely the discoveries of science. Transduction experiments conducted by Hershey and Chase. Through these experiments, they proved DNA is a genetic material. It is a genetic material. So according to them, DNA is the chemical basis of heredity. It is the chemical basis of heredity. Important point. Whereas the physical basis of heredity is chromosome. It is the chemical basis of heredity. Later on on DNA, crystallography techniques conducted by Franklin and Wilkins. Not only these experiments, many experiments helped to understand the DNA structure and chemically analyzed by chemical analysis by Chargaff. So Chargaff chemical analysis, and Pauling's another scientist, Pauling. He proposed hydrogen bonds formation in between nitrogen bases. According to Pauling, in DNA, purines always complementary to pyrimidines. And that too, what he explained, adenine always complementary with thymine by 2 hydrogen bonds. Cytosine always complementary with guanine by 3 hydrogen bonds. So that's the involvement of scientist discovery. Anyway, now detailed description about the DNA. When you go to DNA structure, you know it is a double helical model. According to Watson and Crick, the DNA, double-stranded DNA made up of 2 strands. 2 strands are there like this. These 2 strands are like a twisted ladder. It is like a twisted ladder. 2 strands are there. Each strand is a polynucleotide strand. So how many strands are there? 2 strands. Each one is a polynucleotide. Poly means many, many nucleotides are there. Each strand made up of. Length of the DNA. Length of the DNA is uncertain. Uncertain. Okay. Okay, the two strands are there. These two strands are always anti-parallel. So one is starts with fifth prime, another one is starts with 3 prime. So the two strands are anti-parallel. When you take the diameter of these two strands, DNA diameter is about 20 angstrom units. Remember, diameter is about 20 angstrom units. Whereas the length of the DNA is uncertain. Then when you go for many coils are present, it's a twisted ladder, many coils. Length of the each coil is about 34 angstrom units. Length, it is length clearly. Length of the each coil. And when you observe one coil, how many nucleotides are present? 20 nucleotides. In each coil, 20 nucleotides. It means 10 pairs. In that, what they are saying, adenine is complementary with the thymine by 2 hydrogen bonds. Cytosine complementary with guanine by 3 hydrogen bonds. Always like that, cytosine complementary with guanine, adenine is complementary with thymine, thymine is complementary with adenine, guanine complementary with the cytosine. 1, 2, 3, 4, 5, 6 pairs are over, and again 7, 8, 9, 10. Adenine complementary with thymine. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Like that, 10 pairs are present. Distance between one nucleotide pair to another nucleotide pair is about 3.4 angstrom units. It is a structure. So length of the each coil 34 angstrom units, whereas the distance between one nucleotide pair to another nucleotide pair is 3.4 angstrom units. The backbone of the DNA is sugar and phosphate. So how the backbone is present? Detail we will discuss. How the backbone is present. So look at here. I am taking the ribose sugar. This is the ribose sugar. Second ribose sugar. This is the third ribose sugar. Very important point, observe clearly. Ribose sugar. You know, first, second, third, fourth, and this one is the fifth one. Like that, this one is the fifth one, and this one is the fifth one. Carbon. So nitrogen base, suppose I am taking the adenine clear here. I am taking thymine here. I am taking cytosine here. So three nitrogen bases, adenine, guanines are purines, cytosine, thymines are pyrimidines. For count I am taking three nitrogen bases. This is a ribose sugar. So always nitrogen bases are attached to the first carbon. Okay, these bonds are called glycosidic bonds, you know that one. And here phosphate is added to the fifth carbon, like this, fifth carbon. This bond is called ester bond. This bond is called ester bond. Remember, for glycosidic bond, water can release. For ester bond also, water can release. Not only that one, you know, here I am taking the deoxyribose sugar. Here H and H, here H and H, here H and H. Correct only, na? Whereas here H and OH. OH, here H. So what they are saying, here the diester bond established between phosphate and OH at the third carbon. So this is diester bond. For the formation of diester also water can be released. So you look at here, 1, 2, 3 nitrogen bases. I am taking adenine, thymine, cytosine. 3 nitrogen bases are there. How many phosphodiester bonds are there? 1 to this side. And this is one side. I am going to another side. Look at here. So now reverse order. This is ribose sugar. This is ribose sugar. This is ribose sugar. This is, sorry, deoxyribose sugar. This one all are deoxyribose sugars. Deoxy. So you take this one, fifth carbon. This is fifth. And you take this is the first. Always you know at the first carbon nitrogen bases can be bound. Adenine always complementary with thymine, so their hydrogen bonds are forming. Here thymine always complementary with adenine, here two hydrogen bonds can be established. Here guanine, here hydrogen bonds can be established. So here the fifth one, it means phosphate can be added. Here fifth one, phosphate can be added. Here phosphate can be added. Here also you take this is the third carbon. This is the third carbon. So you tell again phosphodiester, this is OH. So this is the third one, fourth one, fifth one. So here phosphodiester bond, here phosphodiester bond. So here three nitrogen bases, two phosphodiester bond. There three nitrogen bases, two phosphodiester bonds. It means how many nitrogen bases? 3 plus 3, 6 nitrogen bases minus 2, total 4 phosphodiester bonds. So 1, 2, 3, 4. Remember that one. Always minus 2 phosphodiester bonds. So like that, phosphate and sugar are attached by phosphodiester bonds, whereas adenine, thymine, adenine, cytosine, guanine, they are binds by hydrogen bonds. So like that, for the DNA, what is the backbone? Phosphate and sugar. They are linked by phosphodiester bonds. This is the structure of the DNA. Actually, DNA, bDNA is always coils on right-handed side. Only coils on right-handed side. This is the bDNA is the common DNA. Whereas zDNA, that always coils right-hand side and also left-handed side, zigzag manner. It is bDNA, that is zDNA. bDNA is the common DNA. So remember, this one is the nucleic acids, especially DNA completed. Whereas when you go to RNA is formed from the DNA by a process called heterocatalysis. So properties of DNA, first we should know. What is the property? Autocatalysis and heterocatalysis. Autocatalysis means formation of new DNA. It is the older DNA strand. It is the older DNA. It is a new DNA. Formation of new DNA from the older DNA in presence of DNA polymerase. DNA polymerase is called autocatalysis. DNA dependent DNA synthesis. Here in autocatalysis, two strands act as a template. So this older DNA strand, two strands act as template. It is the new DNA. It is a new DNA strand. Remember that one. This is autocatalysis in presence of DNA polymerase. Whereas in heterocatalysis, all the RNAs are formed from the DNA. Remember that one. That may be mRNA, messenger RNA, or rRNA, or tRNA, anything you take. So in heterocatalysis, so actually this is the DNA, two strands. In the two, this one is old strand, this one is also old strand. In that only one strand act as template. Only one strand act as a template, not two strands, which forms RNA by a process called transcription. So synthesis of mRNA from the older DNA in presence of RNA polymerase, is called transcription. After that, this mRNA involved in protein synthesis. That is called translation. Remember that. So both transcription and translation comes under heterocatalysis. Anyway, finally rRNA, mRNA, tRNA, three types of RNAs are present. Very simply we can discuss. mRNA is the messenger RNA, rRNA is ribosomal RNA, tRNA is transfer RNA.